Okay, welcome to lab training. So, I'm just going to go through some of the ins and outs and the lab safety and stuff for this lab. First of all, the fruit flies, a lot of them are still laying down, so you'll want to prop the fruit flies up. And I did notice when I just glanced over there, I didn't look at all of them, but I noticed that there are some dead fruit flies, ones that are stuck in their food that have drowned. So if the students need to redo fruit flies, that's what you should do right off. So when they come in, go get fruit flies, find out who's got dead flies and needs to replenish them, anesthetize them, and have them pick new flies. Then otherwise stack all the rest of them upright and have the students whose flies didn't die, have them count. Now it should still be the original 3, they probably haven't reproduced yet, but they need to take their tallies each week. Alright, so that's the fruit flies. So this lab is food calorimetry, and this is probably the most dangerous lab because they're burning stuff, so you want to make sure you're on top of it. Goggles, you have to wear goggles. I know, it sucks. And the goggles, they should be put out on the table. Clarissa, if you can just put them out on the table, that would be great. If you can't, if you need more, they're in the back cupboard underneath the big fish inside the room. Alright, so let's see. Follow your instructor guide. Make sure you read the instructor guide. It's got a lot of tips to save you from disaster in this lab. Alright, so this is the setup that will be set up on everybody's table. And so what they're going to do is they're going to burn food. Now, I would encourage the students to burn the food all together, burn all the samples and get all the data. And then when all flames are out, then everybody can take their goggles off. So that's the thing. If there's an open flame in the room anywhere, everybody has to be wearing goggles. So even if it's not burning at their table, everybody needs to be wearing goggles, including you, until everybody's burned their food samples. And they're going to burn, what is it? Let's see. They're burning peanut, corn chip, walnut, honeycomb, and oyster cracker. So they got to burn 5 things. So this is how it works. Make sure they tighten up all the bolts and that everything's tight in there. This is the clamp for the test tube, and so I've already placed my test tube in there. Notice that the test tube is all the way down, so most of it's sticking out below the clamp. And that's because if the flame gets big enough, it will melt the rubber or the plastic on the clamp, and we don't want that to happen. And then the thermometer just slips inside the top clamp and just sort of rests on its little thing. It should be almost to the bottom of the test tube, but not touching the bottom of the test tube. If it touches the bottom of the test tube, it's not that big of a deal, but it would be better if it didn't. Alright, so what the students are going to do is they're going to take some foil, and this is going to be what catches any of the ash or the oil that drips out of some of the nuts. So I kind of make it into a little, I don't know, a little bowl-ish. Okay, and so then they're going to put that on the scale, and they're going to zero the scale. Okay, and then we're going to weigh out our sample. Now you want to make sure you use the same foil for the same food sample because you've zeroed it with that particular foil. Oh, I take that back actually. Oh, those are rancid. Hold on. Just kidding. You're going to weigh it with the foil so we can do all the samples at once. It shouldn't matter. Either way, they just got to take into account the foil. Alright, so we've got the foil on there. My foil weighs 1.8 grams. You want about a 1 gram sample. So they're going to pick up a nut. So that one went to 2.3. I'm going to just stick with half of a nut. You want to be able to hold it in the tweezers. So if they get like multiple pieces, they're not going to be able to hold it in the tweezers. So they're going to record their initial 2.3 grams. That's the foil and the nut. Alright? And then you're going to take 10 mils of water. So they're just going to walk over to the sinks and fill it to 10 mils, and you're going to pour that into your test tube. You probably should have done this before you weighed it. Alright, and then we're going to take the initial temperature of the water and make sure they're writing their temperatures down in Celsius, because if they do it in Fahrenheit, it's going to screw it up. Alright, because specific heat is measured in Celsius. So they're just going to measure, I don't know, it's still moving a little bit. So let's say it's 22 degrees Celsius. You write it down, and then from here, goggles on because we're burning things. Okay. And here's a tip, and it's written in the instructor guide. If the students choose to hold the nut like this as it's burning, as the nut burns, it's going to drip boiling oil down the tweezers and onto their fingers. So you just need to be smarter than the fire. So they should be holding it upright and you want as much of the fire to get into the water as possible. So they're going to light it out here because you don't want to be heating the water with the flame, but you want to get it over there as soon as possible. So you're just going to light the food sample on fire, and it kind of takes a little bit for it to get going. Alright, so this one's going, and then you just place it underneath, and remember to hold the tweezers up, and all we're doing is heating the water. So they're just going to leave it like this. The two nuts actually burn quite a bit, so they need to pay attention. So somebody else in their group needs to be paying attention to the temperature. If the temperature reaches 80 degrees Celsius, you want to blow it out. And notice I've got the foil underneath to catch anything that might drip or fall, or if it starts to disintegrate. Okay, once it reaches 80, you want to blow it out, because what happens when water gets to 100? It boils. And so at that point, you're not collecting any temperature change. So we blow it out at 80 to give us a buffer, because it'll keep going up. The temperature, the thermometer rises slower than the actual temperature does. So, and I'm not going to let this burn the whole time because in the interest of time. Now notice how carefully I blew it out. Now what I'll notice students will do is they'll freak out because it gets to 80, and they'll blow it out, and they'll blow flaming peanut all over the papers on the table and all of that. So you want to make sure they're being very careful. So I drop it onto the foil, and then I give it about 10 seconds or so, because the temperature will continue to rise. So they just want to catch that highest point. It's already started to fall, but it got up, whew, I'm gonna choke on smoke. It got up to about 48 degrees. Okay, so I blew it out early. You should let it get up to 80, and then it'll probably climb up to 90 before it actually stops climbing. So they're going to record that on their student guide. Then you're going to take the same foil and then the leftovers, and you're going to weigh it again to see how much mass it lost. So mine says 2.1 grams. So I'm going to write that down. Obviously you would burn it longer, it would lose more. Okay, and that's all in their little student guide in their lab book. They're going to write all those down. And that's all they should do. They should not go through the calculations until they've burned all 5 samples. That'll minimize the amount of time that fire is actively going in the room, and everybody has to wear goggles. So you're going to walk around each table and keep them moving on the 5 food items. Plus it starts to stink, so make sure that they get those done. The walnut is especially flammable, and they will have giant flames licking up, so make sure they're just sort of keeping it controlled. On that note, another thing, there are cubbies over by the door where they should put their backpack and their books and whatever else. The only thing they should have on their table, and even that's pushing it, is their student guide that they're recording the data. And just make sure that they keep that away. We have had somebody blow a food item that landed on a piece of paper and the paper caught on fire. So we want to make sure that we're not doing any of that. The fire extinguisher, just to remind you, is over by the whiteboard. So if you do need that, we don't want the building to burn down. Okay, so notice at the end of this that this test tube is super black. We're not going to use the same test tube again because number 1, the water's hot. Number 2, the test tube's black. It might be hot, so we've given them these little things to pull the test tube off. So you're just going to loosen and make sure your thermometer is good and secure in there so it doesn't fall out and break on the counter. And you just chuck that right there, get a new test tube, a new 10 mils of water, a new starting temperature, and do the whole thing again. All right, for each food item. A couple of things about the waste. So the blackened test tubes, we're just going to throw them away because it's too hard to get the soot off. So all the waste procedures are written on the board behind me, so you just take a look at those. The test tubes are going to go into glass disposal, so it's just a box that says glass on it. Just make sure they get everything in there. If you break any glass on the table, that also needs to go into the glass disposal. The food samples can just be thrown away in the trash. Have them wrap them up so it doesn't get too stinky in here. Aluminum foil trash, obviously. Okay, so that's the first part of the lab. Let me make sure I caught all of the... Oh, and I don't need to be wearing this anymore. And you're all going to have goggle face by the time you leave. It's going to be awesome. Let's see, especially important. So when holding the food samples, I talked about that. If burning the food, if the food drops, so sometimes it'll disintegrate and pieces will drop, or the whole thing will drop, tell them not to try to pick it back up with the tweezers while it's burning, because you're losing tons of the heat because it's still on fire. So if the burning sample drops to the foil, like you were holding it, it drops down, have them just pick up the foil and place the burning sample right back under there until you need to blow it out. Okay, so don't have them waste any time trying to pick it back up. That's another thing. If the water gets to 80, blow it out. We got that. Blow it out carefully. And then do not blow the food samples, because you will blow burning debris all over everything. Okay, so once they do that, they're going to go through. Now, this is the only lab where the homework assignment is not a separate thing. it's actually embedded in the lab. Most of the time they do the lab procedure and then there's a homework assignment sheet. This one is just built right in. So after they finish that, on page 2 of their student guide, it goes through the calculations of how to figure out calories at the very end of that table. So have them follow that procedure. And calories at the end of the table, it's a big C calories. So remember, little c calories is the amount of energy it requires to raise 1 gram of water 1 degree Celsius. But the big C calorie is a kilocalorie. That's why at the end of those calculations, they divide by 1,000. And they're going to be getting something along the lines of 0.6, 0.8 calories per gram, which is really low because it should be more like around 6. But remind the students that not all of the energy, don't tell them this, let them struggle with it. But when they are like, why is my number so low? Remind them that where did all the energy go? I mean, a lot of the energy went into heating the test tube, heating the air. You're not catching it all. This is a very inaccurate setup to do this lab, and we usually capture about 10% of the true amount of energy released. Now in the real world we use a bomb calorimeter, so they actually put the food inside and it burns completely enclosed underneath the water. Yeah, so they're gonna go through the calculations and then they're gonna answer some questions, and all of the answers are written in your instructor guide, so make sure you read through that and understand what you're talking about there. So you're going to let them work on that. Probably to burn the food's only going to take 15-20 minutes, and then they'll work on the homework questions for another 20-30 minutes or so, and then you're going to do the second part of the lab. So make sure you're watching the clock so you don't let them just go on this forever. If they don't finish the homework questions, they can come back to it later, right? Because they just have to attempt the homework before they leave. Okay, so I'm just going to check and make sure this is still recording. Okay, so we kind of stink to do this whole presentation and then realize it wasn't recording. All right, so part 2 of the lab, it tells them they're going to do homework questions part 1 and part 2, and then it tells them to stop and wait for a class discussion. And so that's when you are going to talk about macromolecules. So you are introducing macromolecules to them before we talk about it in lectures, so you want to make sure that you've read through this and you do introduce it, because we're counting on you to have introduced this already. All right, so you're going, and it's in the instructor guide, it kind of walks through, you know, they'll notice that some of the foods like the peanut and the walnut that have a lot of fat content, they burn hotter and they release more calories than like the oyster cracker that's almost hard to get burning at all, and the honeycomb. And so you ask them, you know, why are these different? Let them struggle with it, practice the things we talked about in the training, wait time, having them talk to their group members, that kind of thing. And then you're going to introduce carbohydrates, lipids, and proteins. And you can see on there for carbohydrates, you're going to talk about the difference between a simple sugar and a complex carbohydrate. You don't need to go into the nitty-gritty details of the chemistry. You just need to talk about how carbohydrates are made out of individual sugar molecules, and you can have simple ones that are 1 or 2 together like a disaccharide, or you can have these long chains like starch or cellulose. So that's really all you need to introduce there, and then you're giving them the purpose. So the purpose of carbohydrates is mainly energy, but it can also be structural, like cellulose, that's the cell walls of plants. Okay, and then lipids, you're going to talk about those as not being polymers. They're just a giant molecule altogether. So they're long chains basically of just carbon-carbon bonds. And the main purpose of them is for cell membranes, hormones, padding, insulation, and they can also be used for energy. They have more calories than carbs do. And then proteins, introduce them as a polypeptide made of amino acids, and that they're mostly structural and functional, like an enzyme or something. So on their student guide, they have a spot to fill in that, but you're going to do this in a way that don't just rattle this off to them and tell them. Ask them, what's a carbohydrate used for? Why do we have carbohydrates? Can you think of different kinds of carbohydrates? And have them come up with all this. They will know most of this just from living on planet Earth and eating food. So you don't have to just tell it all to them. Make it an interactive discussion. Same thing with the lipids and the proteins. And then once they've done that, then they're going to try to figure out what macromolecules are contained in things. And so Clarissa, I need to tell you, so I noticed you made the starch solution and you made the glucose solution, but what we actually need for, and you're probably just waiting to do this, but what we need at the lab tables on this week's lab is each table should have a bucket like this with 4 different of these little tiny beakers. They're the little 50 ml beakers, and they should have the juices in them. So there should be a starch solution, a glucose solution, an onion solution, and a potato solution. Now, TAs, probably a good idea is to go ahead and fill them up at the beginning of your lab. So if you, Clarissa, can just have them set out like this. The other thing I have in here is iodine and Benedict's. Okay, so if that's just set out on the tables, then the TAs, when you guys come in, the very first thing you can do is come and distribute the juices into the little Erlenmeyer flasks for the students, because the onion juice and the potato juice need to be refrigerated each evening. So if you're the last lab of that day, and even at the end of your lab, if you can put the bigger solutions into the fridge, that would be great. You can put it in the little black fridge right inside the door, or if there's not enough room in there, you can put it in the bigger white fridge. So you want to put the big solutions into the refrigerator, and it wouldn't hurt to put the starch and the glucose in the fridge too. I'm going to put them in there before I leave today. But the onion and the potato, for sure, they'll go rancid. So if you can put the big solutions away, you can leave the little ones out all during the day. So if the students don't use it all, we can just replenish it from the solutions. But if you are the last lab of the night on Tuesday night or Wednesday night or Thursday, I think it's like during the day, go ahead and gather up all the little tubes and put them all in a refrigerator so that they don't sit out overnight. So that if you're the first lab in the morning and you're looking for all of the little Erlenmeyer flasks, they will be in the refrigerator along with the master solutions. I hope that made sense. If it didn't, send me an email. The other thing we will need, Clarissa, on each of the tables is inside each of the tubes we need one of those little plastic pipetters. They're down underneath in the cupboards. So they're going to use the plastic pipetters to suck it out and put it into test tubes. And then just put a piece of tape on it and label each one. Okay, so it's onions, potatoes, sucrose, and starch. Okay, so that's what we have here. So the second part of the lab, they're using these solutions. So the starch solution and the glucose solution are your controls, right? So they're going to have those, and then they're going to have potato and onion. So Clarissa, when you make the potato and onion, you're literally just going to chop up the potato into smaller pieces, throw it into the blender, and blend up one potato for a whole thing of this. Okay, and the same thing, one onion for a whole thing of this. Now, there's going to be chunks floating around in here, so you want to encourage the students to sort of shake it up before they put it into their flask, and likewise swirl their flask around before they suck it up and put it into their test tube. Like the starch, for instance, I just shook it up, but the starch will all settle at the bottom, and so if they just suck the water from the top, they're going to be like, oh, there's nothing in here. Yeah, there's nothing in there. They have to get the starch back into solution because starch is not water soluble. Okay, same thing with the potato and the onion. Make sure they're shaking it, or they're taking their flask and they're mixing it around before they suck it out and use it in their tube. And so basically what they're going to do is they're going to test for starches and they're going to test for sugars. So when they start this portion of the lab, you as TAs, so Clarissa, this is another thing we need. We need 1 liter beakers for each of the hot plates. So that when they start this portion of the lab, you're going to fill that 1 liter beaker probably only about halfway, maybe a little less than halfway, with water. And then you're going to put it on the hot plates and you're going to put them on high and start them boiling. Because by the time they get to the Benedict's tests, that's the blue stuff, it needs to be boiling. And you want to have it about halfway so that they can stick a test tube down into the beaker and the water doesn't submerge over top, because that's bad. All right, so they're going to test for, let's see, if we look at, we have a little discussion. Okay, so procedures part 2 on their student guide, they're testing for starch. So iodine reacts with the bond between sugar molecules in a starch molecule. It actually gets like somehow integrated into the secondary structure. And so what they're going to do is they're going to fill up their 4 test tubes and label them. Oh, that's another thing, Clarissa. There are some wax pencils in one of those drawers back there. Each table needs a wax pencil so they can label their test tubes. I think they're red. They might be black. All right, so they're going to fill up their test tubes. They're going to label them. Then they're going to drop iodine. And it doesn't need very much. They're going to drop the iodine in, and then you're going to teach them, guys, how to mix a test tube. I should not see any of this going on, putting the thumb over top. Now, none of this is dangerous, but this is not a good habit to get into. So the way to mix a test tube is to hold it at the top and then drum it with your fingers, and that will mix it really well. Drum it hard, okay? So they're going to mix it. Now, if there's starch present, it will turn black. If there's not starch present, it will stay the yellowish brown. But what I noticed with the starch especially, or I'm sorry, with the potato especially, is that it's not fine enough for it to, the whole thing to go black. So they might say, oh, a potato doesn't have starch. Have them look at the bottom of the test tube and they'll see little black pieces down at the bottom, okay? The glucose should not turn, the onion should not turn, but the potato should. So you might ask them, so how come a potato has starch but an onion doesn't? That's weird. What is the starch doing for the potato? Now remind them that the potato doesn't put starch in itself for us to eat it and enjoy, right? They don't have us in mind when they're doing this. So what are they using the starch for? And they can have a little discussion about it. So it turns out that starch is food storage. Just like we store glycogen in our muscle cells, potatoes store starch. So then you might ask, well, an onion, doesn't an onion store for the plant also? It does. It just stores it in a different way. It's going to store it as simple sugars instead of starches. And that's why an onion is sweet, relatively speaking, to a potato. All right, so they're going to test for starch in all of those. And the answers are in your instructor guide. And then for procedure part 2, they're going to test for sugars. So Benedict's is a really cool thing. It reacts with simple sugars. So if it's starch, it's going to stay blue. But if it's a simple sugar like a monosaccharide or a disaccharide, it's going to turn various shades. You can look at the table in their student guide, various shades of yellow, orange to red. But it doesn't change unless it's boiled. So they're going to set up their same set of test tubes. They're going to drop in Benedict's. So they should put in, about one mil of the, and that doesn't have to be exact, but 1 mil of the solution. And then they're going to add an equal amount of Benedict's. So this will probably have to be filled again about halfway through the week. And so I noticed Clarissa has put the big container of Benedict's. That is the wrong iodine. I will grab the right iodine and put it there. So the iodine and the Benedict's, so you can refill. So Clarissa, the iodine is not the powdered stuff, it's the Lugols. So it's L-U-G-O-L. And it's a big, tall one that's a liquid. So I'm going to put that out there. So anyway, they're going to pour that in there, but you may have to refill these. So you've got the solution over there. There's little funnels in back in the glassware cabinet if you need to use a funnel to fill them. Okay, so don't call Clarissa down here to refill tubes. You guys can do that. So anyway, so you're going to pour it in there and then you're going to put it into the boiling water bath. It has to be actively boiling. If you do not see bubbles rising up from the bottom of the beaker, it's not hot enough and it won't react. Okay, so don't let that fool you. It's got to be an active boil. So they put them in there. Just make sure they've labeled them with their group number and then what it is, because they're all going to get mixed up in the beaker. And it says 5 minutes in the lab, but they'll start to see a change after about 1 minute, and it will be a definite, obvious green to yellow to orange to red. Make sure they're using these to get it out, because students forget that, hello, I just put it in boiling water, it's hot. Okay, and then you're going to get broken glassware because they're going to pull it out and go, ah, and drop it. So make sure they're using these to get it out of there. And they can put it right back into their test tube rack. It won't hurt the test tube rack. So that's the part 2. Then they do some homework questions. The rest of part 2, they're going to test more with the Benedict's. So in this case, they're taking a little chunk of paraffin wax. So this is parafilm, it's paraffin wax. So you're just going to cut off a square. They kind of come in little squares with a pair of scissors. So we'll probably need a pair of scissors, Clarissa. There are some in the back in the drawer. So they're going to cut off a little square, pull off the paper backing, and then they're going to chew it like gum. It's just wax. They can chew, chew, chew. And as they're chewing it, they are producing saliva. That's what helps them produce saliva. So one tube's going to have just plain starch in it. The other tube is going to have starch with spit. So they're going to just put the test tube up to their mouth. It's disgusting. Put the test tube up to their mouth and put as much spit into the tube as they can possibly muster. Now, once they've spit into the tube, it is considered a biohazard. So the person donating the spit for the group has to be the one that handles that tube the rest of the time. Okay, so they're gonna add the Benedict's to it. They're gonna put it in the hot water bath, and they're gonna collect it at the end. Okay. I noticed on the board it said the paraffin wax can go in the normal trash. I'm gonna change that. The paraffin wax should be spit into the biohazard glass also, because they chewed on it. All right, and then the spit test tube goes into biohazard glass. So you'll notice there will be 2 glass containers out here. One of them is just a normal box bag. The other one is a red bag. The red is the biohazard. The normal one is not biohazard. So make sure all of the burnt test tubes that you did for the food calorimetry, you empty the water out. That can just go in the normal non-biohazard. We don't want to fill up the biohazard with these test tubes because that would be a waste. because they have to do extra things to the biohazard waste. Let's see, so that's that. And then at the end, you're just talking about tween, which is a detergent. You don't actually do anything with this, but tween has a hydrophilic head and a hydrophobic tail. And so it acts like soap, so it will surround lipid molecules and break them up. It emulsifies them, so it actually makes them into smaller little droplets. And if we looked at it under a microscope, you could see it, but we're not going to do that. So just have them sort of walk through the questions. It's outlined very well in your instructor guide, in case you want to know. In the instructor guide, I think all of the terms you're supposed to introduce are in bold, so make sure you get all of those down. Oh, one other note, on the spit thing, it has to incubate for at least 30 minutes, so you might encourage the students to have one of their group members break off when they start doing these procedures and go ahead and get the spit going. Like direct them to that part and say, hey, you ought to get that spit going because it's got to go for half an hour. And then if they have extra time when they finish, they should be working through the homework questions and making sure they understand all of those. This lab has a ton of garbage, so make sure you make your students clean up after themselves, like wipe down the tables, get some paper towels, don't leave water or soot or anything all over. Also, you'll end up with soot all over your face like I have. Make sure they throw away all their garbage, make sure all the glassware goes away, and that they leave the table exactly as they found it, with everything stacked in the middle and looking really nice. Let's see. Okay, so I talked about potato and onion juice being refrigerated, goggles including you as long as flames are going, waste things are on the board for you, make sure all papers, books, and backpacks are off the tables while flames are going, check the flies and redo them if necessary. Also have them do their counts for week 1. I think that that's everything. I hope that I covered this well enough. All the food samples are over there on the table. The Sharpies are over there for counting the fruit flies. Make sure they record what color they did their dots with this week. And if you forget about that, read your instructor guide on their setup. So yeah, here's a clamp that's been burned, and you can see it looks disgusting. So make sure that they're keeping the flame well below the clamp. Okay? I think that that's everything. So if you have questions, please email me. Otherwise, we are good to go. Oh, and they're going to be turning in their library lab this week, so make sure you collect that. And there should be, you have the guidelines, I think, in your packet, so you're just going to grade them. Really, it's just a completion thing. You're not going to be nitpicky. If they did exactly what the library lab had them do, if they've got their annotated bibliography and all that, they're going to get 10 points. Okay, just make sure they follow directions. Yeah, and there's no quiz this week. There will be one next week. All right? Okay, I'm done. Okay.